Hi everyone, welcome to my A-Level Biology Extended Lecture Series. In this lesson, we will explore X chromosome inactivation. Let's get started. So what is X chromosome inactivation, or known as XCI? So X chromosome inactivation is a random, irreversible process that occurs in each somatic cells of a female embryo. So be careful, only occur in female. So in female who are heterozygous for X-linked gene, only one of the two X chromosome remain active in each cells. We do not know why, but generally we know that particularly in mammal, a female inherits two copies of the X chromosome, one from paternal gamete, another one from maternal gamete. So in this case, instead of two copies of X chromosome remain active, due to X chromosome inactivation, one X chromosome will be selected, and this is random and irreversible when this selected X chromosome is inactivated. So this means that approximately 50% of cells express one allele, while the other 50% express the alternative allele. So because in the heterozygous conditions, so two alleles that's located on the X chromosome, the cells will express 50% and another 50% will express the another allele. So this phenomenon can sometimes lead to visible phenotypic effect, especially when X-linked gene influence their observable trait. So let us look at a diagram to show how X chromosome inactivation actually takes place. So we can see that cell number 1 is the newly formed zygote. So this newly formed zygotes undergo few rounds of mitosis to form embryo. So in these cells, you can see that at first they have two X chromosome, right? Okay, so one of them is red color, another one is dark blue. However, when come to embryo stage, the random and spontaneous, random selections of one of these X chromosome occur, and this X chromosome is inactivated and form a body. So what will happen next? The descendant of these cells will have only one active X chromosome. So if you look at the cell left-hand side descendant, all of them will have red X chromosome. However, on the right-hand side, the descendant of the cells will have dark blue X chromosome. And this is obvious when some phenotypes depends on, or some phenotype depends on the genes that they are located on X chromosome. So some gene controlling coat color in cat and mice are located on X chromosome. So in female mammal, they are heterozygous for this gene. X chromosome inactivation will lead to patches of tissues where different alleles are expressed, resulting in patches, or we call it as a variegated fur pattern. So this is clearly seen in a tortoiseshell or calico cats, where patches of black and orange fur reflects the random inactivation of one of the X chromosome in early development. So how this actually happened? So remember what we said, that during embryo stage, we have, let's say we have two type of cells. One express the orange color, another one express the black color. Okay, so if X chromosome inactivation take place, so the descendant of one cells, will only express orange color. When mitosis take place, the whole group of cells, as say that descendant, will always follow back the original. So the original one now already inactivated the black color allele. So therefore, only orange color allele is expressed. So the descendant of cells, all of them express the orange color allele that located on the active X chromosome. So another group of cells express black color allele, okay, or black fur color allele in the descendants of the cell. So therefore, when you look at this kind of cat, immediately we know it must be female because X chromosome inactivation won't take place in male. Correct or not? Right? Because the gene can only be found on the X chromosome, but not Y chromosome. So X chromosome inactivation occurs early in the embryonic development when only few cells are present. So once inactivated, the same X chromosome remains inactive in all descendant cells through mitosis, producing clones of the cells with the same active X chromosome. So if this X chromosome carry orange color allele, so the descendant of cells will express orange color, so give us a whole patch of cells that produce orange color pigment. While on the other hand, another group of cells produce or with black color allele. So therefore, the whole patch of cells express black color fur. So each fur patch represents a clone of cells expressing the same allele, leading to the visible mosaicism or patches of color. In humans, the effect of X chromosome inactivation may not always be visibly detected. For example, we have red-green color blindness. It's caused by a mutant allele on X chromosome that affect the cone cells that detect the color, okay, in the retina. So a heterozygous female may have patches of defective cones, but the presence of the normal cone in other patches usually result in normal color vision. Is sufficient even though half of it, right, or half of them are functioning. In trait where the gene product is secreted into the bodily fluid, such as hemophilia, mosaicism is not usually apparent. You won't be able to detect it. For example, in a heterozygous female for the hemophilia allele, about half of her cells produce functional factor 8, the another half of the cells produce insufficient or may not produce factor 8. So this amount usually sufficient to maintain normal blood clotting, even though only half the cells produce the functional factor 8. And with that, we have come to the end of this lesson. Hope you found it helpful and easy to follow. If you have any question, feel free to drop them in the comment section. See you in the next lesson. Have a great day.